Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University, School of Continuing Education, SOCE, Master's Degree Programs, Master of Science, Food Nutrition, MSCDFSM, Revised First Year MFN 002 Nutritional Biochemistry Unit-10 Vitamins and Minerals 10.1 Introduction. We have already studied the chemical nature of the various vitamins earlier in Unit 3. We have also come across vitamins when we have studied the various metabolic pathways, Units 6, 7 and 8. The chemical reactions occurring in our body are catalyzed by protein molecules called enzymes. We have read about the characteristics of enzymes in Unit 4. We learned that many enzymes are active only when other molecules called cofactors are present. In most cases, these cofactors are vitamins. If these cofactors are absent, the enzymes will not act and may lead to the occurrence of disease. Thus, vitamins give protection to the body against diseases. In order to understand the functions and metabolism of vitamins in the body, you must become very familiar with their structure and properties. Hence, at this point, you should revise Unit 3. You would have read in that section that vitamins may be classified as fat or lipid, soluble or water soluble. We will continue with this classification in this unit and study about the metabolism of vitamins. Since vitamins play a vital role in human metabolism, it is but natural that if these micronutrients are deficient, absent in our diets, metabolism will become deranged, disordered. This will lead to a diseased condition accompanied by clinical symptoms. The various vitamin deficiency diseases are discussed in the course Public Nutrition in Units 3, 4. The disease is cured or prevented only by restoring the intake of the vitamin to the diet. The next part of this unit focuses on minerals. Minerals are inorganic elements and hence are distinctly different in chemical nature from the other major four nutrients, carbohydrates, proteins, lipids and vitamins, which are organic compounds. Most minerals, except sodium and potassium, form salts and other compounds that are relatively insoluble. Hence, they are not readily absorbed and most ingested minerals are excreted in feces. We shall learn about the metabolism of minerals in this unit. Objectives: after studying this unit, you will be able to name the storage form of the fat-soluble vitamins, identify the active form of the vitamin, describe with chemical reactions the biological role of each vitamin in the body, and discuss the biological role of different minerals in the body. 10.2 Vitamins. Vitamins, as we all know, are organic compounds required in very small quantities for a variety of biochemical functions and which generally cannot be synthesized in the body and therefore must be supplied in the diet. This definition clearly distinguishes vitamins from inorganic ions which are also essential for life. Further, unlike the macronutrients, carbohydrates, proteins and fats, which are required in large amounts, vitamins are needed in only minute amounts. Apart from some exceptions, all animal species require the major vitamins preformed in the diet due to inability to synthesize them from other food constituents. However, in some instances, certain precursors present in the diet are sufficient since these precursors can be converted to the vitamins in the body. One good example of this is the pigment beta-carotene, which you may recall reading, is converted to vitamin A in the body. The vitamins have been named using English alphabets, e.g. A, B, C, etc. In addition, some vitamins are also commonly known by specific names. A few vitamins are not by any alphabet but are referred to only by specific names. Further, we have also studied that they are classified into two categories, namely water-soluble and fat-soluble vitamins. In the subsequent section, we shall study about the different vitamins within each of the two groups. The vitamins after absorption undergo changes in their chemical structure. They become functional only after undergoing these structural modifications. Hence, this is referred to as the active form of the vitamin. Let us get to learn about the metabolism of different water and fat-soluble vitamins. We begin with the fat-soluble vitamins. 10.3 Fat-soluble vitamins. Fat-soluble vitamins are apolar, lacking in ionizable groups, hydrophobic, water-repelling or lacking in water affinity, compounds that can only be absorbed efficiently when there is normal fat absorption. This means that bile, which helps in digestion and absorption of various dietary lipids, is also necessary for absorption of these lipid-soluble vitamins. 10.3 Fat-soluble vitamins. Fat-soluble vitamins are apolar, lacking in ionizable groups, hydrophobic, water-repelling or lacking in water affinity, compounds that can only be absorbed efficiently when there is normal fat absorption. This means that bile, which helps in digestion and absorption of various dietary lipids, is also necessary for absorption of these lipid-soluble vitamins. Hence, apart from low dietary intake, conditions affecting the digestion and absorption of the fat-soluble vitamins such as steatorrhea, fatty diarrhea, and disorders of the biliary system can all lead to deficiency syndromes associated with that particular vitamin. They are transported in blood as constituents of lipoprotein molecules, molecules containing lipid plus protein, or attached to specific binding proteins. The fat-soluble vitamins are stored in the body if their intake is more than the recommended amounts. This is a desirable feature. It ensures that these vitamins from storage become available to the body when dietary intakes are reduced. At the same time, excessive storage of the fat-soluble vitamins leads to signs of toxicity, which in many instances can be extremely severe. As you already know, there are 4 fat-soluble vitamins. These include A, D, A and K. Now we will discuss each fat-soluble vitamin individually. Revise the structure of the vitamins as given in Unit 3 earlier, before going on to the biological role. 10.3.1 Vitamin A. Vitamin A occurs only in animal tissues. But many plant tissues contain substances, precursors, which can be converted to vitamin A in the body. These are the yellow or red-colored carotenoid pigments found in plants. These include carotenes, alpha, beta and gamma, and the related compounds, cryptic xanthan, known as provitamin A. You have already learnt earlier in Unit 3 that the active forms of vitamin A are retinol having an alcoholic group, CH2OH, retinol, also called retinaldehyde, having an aldehyde group, CHO, and retinoic acid having a carboxylic group, COOH. Vitamin A is stored as retinol palmitate, ester of retinol and palmitic acid. This ester is stored in the liver as a lipoglycoprotein, i.e. fat-soluble vitamin plus carbohydrate plus protein. For transport to the tissues, the complex is hydrolyzed and the retinol is bound to a protein called retinol binding protein, RBP, and secreted into the plasma. Retinoic acid is transported in plasma bound to albumin. Once inside the cell, extrahepatic, retinol is bound by another protein called cellular retinol binding protein, CRBP. Let us next learn about the role and the mechanism of action of vitamin A in our body. You may have already learnt about these functions in the Advanced Nutrition course in Unit 7. Here we shall look at these functions from the nutritional biochemistry point of view. Functions of vitamin A. One of the functions of vitamin A which has been known for a long time is the role it plays in normal vision. To understand this function, you must revise at this point the physiology of vision discussed in the course Applied Physiology in Unit 10, Physiology of Special Senses. You may recall reading in this course that retina has 2 kinds of cells, rods and cones. Rods help us to see in dim light while cones function in bright light. Rods and cones contain light-sensitive protein called opsin which combines with retinaldehyde as the prosthetic, additional, group to form a complex called rhodopsin or visual purple in rods and iodopsin in cones. Figure 10.1 illustrates light activation of rhodopsin. Let us now see the exact mechanism by which the rods function in dim light. You have seen in Unit 3 that vitamin A exists in several isomeric forms. These isomers have the same number of atoms and the same kinds of groups, but differ in the arrangement of groups around the carbon atoms. Thus, retinaldehyde exists in 2 isomeric forms, all trans or 11 CIAs. In all trans isomer, all the double bonds have groups on both sides of the double bond. In 11 CIS isomer, the double bond between carbons 11 and 12 has groups on one side of the double bond. In rhodopsin, the amino acid lysine in the protein opsin forms a complex with 11 CIS retinaldehyde and this helps us to see in dim light. When we look at bright light, a number of complex biochemical changes take place and a nerve impulse is generated. There is isomerization of 11 CIS retinaldehyde to all trans retinaldehyde. This leads to a conformational, structural change in opsin and different isomeric forms result. When bright light falls on rhodopsin within 10 to 15 seconds, 11 CIS retinaldehyde is converted to all trans retinaldehyde and the complex photorhodopsin is formed. Next, bath-rhodopsin is formed within picosecond of illumination. This is followed by a series of conformational changes and ultimately metarrhodopsin 3 is formed. In the final step, this molecule is hydrolyzed to all trans-retinaldehyde and opsin. This is also called bleaching of rhodopsin. So bright light markedly depletes the stores of rhodopsin in the rods. Figure 10.1 explains light activation of rhodopsin just discussed. Figure 10.1. Light activation of rhodopsin. So what happens when this person leaves a well-illuminated room and enters a dimly lit room? Obviously the person is unable to see. Why? Because rhodopsin in the retinal rods has been hydrolyzed into its two components, opsin and 11-cis-retinaldehyde, which is isomerized to all trans-retinaldehyde. For vision to be possible, two events have to take place. Firstly, all trans-retinaldehyde in the rods has to be first isomerized to the specific 11-cis isomer and then it has to combine with the protein opsin to form rhodopsin. This accounts for the well-known fact that an individual has difficulty in seeing on entering a dimly lit room from a well-lit place. After several minutes, during which time rhodopsin is synthesized, vision improves to the point that one may marvel at one's inability to see a short time previously. Are you in the habit of entering a cinema hall after the show has started? If so, you would be very familiar with this situation where you feel totally blind on entering, but within a very short time you can see everything. This is called dark adaptation and the time taken to achieve it is called the dark adaptation time. For rhodopsin to be reformed, all trans-retinaldehyde is first converted to all trans-retinol, which is then isomerized to 11-cis retinol. This is then converted to 11-cis retinaldehyde, which combines with opsin. There is a loss of vitamin A in the photochemical, light-induced reactions involving rhodopsin. If the blood is not well supplied with vitamin A, as seen in vitamin A deficiency, the time required for rhodopsin synthesis is lengthened or the total synthesis may not reach optimum quantities. Under such circumstances, dark adaptation time is subnormal. When the condition becomes chronic, it will lead to night blindness. This visual process starts from rhodopsin and comes back to rhodopsin. Hence it is commonly called the visual cycle or rhodopsin cycle, as illustrated in Figure 10.2. Figure 10.2. Visual cycle, rhodopsin cycle. E. Vitamin A takes part in the control of cell differentiation, forming different types of cells for different tissues, and turnover, growth. All trans-retinoic acid and 9-cis retinoic acid regulate growth, development and tissue differentiation. They have different actions in different tissues. In this function, retinoic acid acts like a steroid hormone. It binds to nuclear receptors that bind to specific regions of DNA called response elements. This then causes expression of that gene, i.e. synthesis of mRNA, transcription takes place. Ultimately, this will lead to the synthesis of a specific protein required for growth. We will read about how steroid hormones act in Unit 11. E. Retinoic acid participates in glycoprotein synthesis. This may partly explain how retinoic acid helps in promoting growth and differentiation of tissues. It is believed that retinoyl phosphate functions as a carrier of oligosaccharides, carbohydrate containing a few monosaccharide units, across cell membrane. This oligosaccharide is used for synthesis of glycoproteins, carbohydrate plus protein, which in turn are necessary for normal growth and also for mucus secretion. Mucus is the lubricant coating the epithelial cells and it contains a glycoprotein, mucin. This is the reason why in vitamin A deficiency there is a reduction in mucus secretion and keratinization, thickening, of epithelial tissues of eyes, lungs, gastrointestinal and genitourinary tracts. Fissures, cracks, readily develop in such epithelial tissues and make them more susceptible to bacterial invasion and can lead to entry of microorganisms causing various infections, particularly in children. Good vitamin A status in the body prevents illness and hence vitamin A is called the anti-infective vitamin. Additionally, since vitamin A has an important role in differentiation of immune system cells, even mild deficiency leads to an increased susceptibility to infectious diseases. Synthesis of retinol-binding protein is also reduced during infections. This decreases the circulating vitamin and therefore there is a further impairment of immune responses. IV, retinol and/or retinoic acid is required for the synthesis of the protein transferrin which is needed for transporting iron in blood. This is the reason why vitamin A deficiency can lead to anemia. V. Both retinoids and carotenoids have anti-cancer activity. This again is attributed to the role of retinoids in cell differentiation. V. Beta-carotene is an antioxidant, effective at low oxygen concentrations. Compounds like unsaturated fatty acids with double bonds are very susceptible to oxidation, peroxidation, giving rise to what are called free radicals, about which you may recall reading in the last unit. These free radicals, as you know, are considered extremely toxic since they damage tissues and cause cancer. Beta-carotene plays a role in trapping peroxy-free radicals and preventing development of cancerous tissues. Next, let us look at the mechanism of action of vitamin D vis-a-vis its functions. 10.3.2 Vitamin D. You have already studied earlier that there are two forms of vitamin D called D2, ergocalciferol, and D3, cholecalciferol. They have a steroid structure containing cyclopentanoperhydrophenanthrene ring. Look up Unit 3, subsection 3.4.2 for the structure. Vitamin D2 and D3 are formed by irradiation of plant sterol, ergosterol and animal sterol 7-dehydrocholesterol, respectively. For vitamin D to be physiologically active, it has to be converted into the active form. In the liver, cholecalciferol, also called calciol, which has been synthesized in the skin or derived from food, is hydroxylated to form the 25-hydroxy derivative called calcidiol. This is then bound to a vitamin D binding globulin, which is the main storage form of the vitamin. On release from liver, it enters circulation and goes to the kidney. Here, calcidiol can undergo two reactions: hydroxylation in position 1 to form 1,25-dihydroxycholecalciferol, also called calcitriol. This is the active form of the vitamin. B. Hydroxylation in position 24 to form 24,25-cholecalciferol, also called 24-hydroxycalcidiol, which is an inactive metabolite, intermediate. Ergocalciferol undergoes similar hydroxylation to give ercalcitriol, active form. These structures are given in Figure 10.3. Figure 10.3. Metabolism of vitamin D. The active form is then transported to the target tissue for exerting its effect, which are discussed herewith under the heading Functions. Functions of vitamin D. Vitamin D plays an active role in calcium metabolism. Thus, it helps in the control of calcium homeostasis, i.e., maintaining normal calcium levels in the body. In fact, vitamin D not only regulates calcium homeostasis, but its own metabolism is in turn regulated by calcium homeostasis. Further, calcium metabolism is interlinked with the metabolism of phosphorus in the body. Hence, metabolism of vitamin D, calcium and phosphorus are all interconnected and interdependent, as you will find out while learning about these three compounds. As already mentioned above, the principal function of the active form of vitamin D, i.e. 1,25-dihydroxycholecalciferol, calcitriol, is to maintain the plasma calcium concentration at desirable level. It achieves this in three ways. It, I, increases intestinal absorption of calcium, E, reduces excretion of calcium by stimulating resorption in the distal tubules, in the kidney, and E, mobilizes bone mineral by dissolving calcium deposited in the bone matrix. How does vitamin D increase intestinal absorption of calcium? For this, the action of vitamin D is like that of a steroid hormone. You will read about steroid hormones in detail in Unit 11. We will have a brief look at it here. When calcitriol from blood enters the intestinal mucosal cell, it binds to a special protein receptor in the cell cytosol. This complex is transported to the nucleus, where it binds to a specific DNA. This stimulates the enzyme RNA polymerase II. As the name suggests, this enzyme brings about the synthesis, transcription of a specific mRNA, messenger RNA. The mRNA is transported to the cytoplasm. Here it attaches itself to a ribosome and brings about the synthesis, translation, of a specific calcium-binding protein called calbindin. 1 atom of calcium is bound per molecule of protein. When food is digested, this protein enters the intestinal lumen, binds to calcium ions and transports the bound calcium. This then enters the bloodstream and raises a lowered blood calcium level. The role of vitamin in calcium homeostasis is shown in Figure 10.4 AD. You would realize that calcitriol works along with parathyroid hormone, PTH. This hormone is secreted by the parathyroid gland when there is a low serum calcium level. Figure 10.4a. High PTH level promotes the formation of calcitriol, 1,25-dihydroxycholecalciferol, which, as explained above, promotes calcium absorption in the intestine. In bone, calcitriol and PTH act synergistically to promote bone resorption, demineralization. Finally, PTH and calcitriol inhibit calcium excretion in the kidney by stimulating calcium resorption in the distal renal tubules. When serum calcium levels are high, reverse reactions take place as shown in figure 10.4b. Production of PTH is blocked. Low PTH levels increase the formation of the inactive 24,25-dihydroxycholecalciferol. This leads to an inhibition of bone resorption while calcium excretion is enhanced. Thus we see that vitamin D activity is closely linked with that of PTH. Additionally, when there is a high serum calcium, the hormone calcitonin, CT, is secreted by the thyroid gland. CT acts in the kidney and increases the excretion of calcium, thereby lowering serum calcium levels. CT also inhibits bone resorption. Figure 10.4c and d illustrates the pathway of calcium metabolism when vitamin D is low and in excess respectively. Figure 10.4, Vitamin D and calcium homeostasis. 10.3.3 Vitamin E. Vitamin E is the generic name for two families of compounds called the tocopherols and tocotrienols. Both of these are present in several isomeric forms. You have already learnt about the structure and properties in Unit 3. Look up these structures once again and then move on to the functions discussed herewith. Functions of vitamin E. Vitamin E is a natural antioxidant. Since vitamin E is fat-soluble, it accumulates in lipoproteins circulating in blood, cell membranes and fat deposits. Without vitamin E, cell membranes, active enzyme sites and DNA, nucleic acid, are less protected from free radical damage. Well, what are free radicals? As you have already learnt about them in the last unit, you know double bonds are very reactive. Hence, they can easily react with oxygen to form peroxides. Peroxides are formed when the double bond between two carbon atoms is replaced by two oxygen atoms. The polyunsaturated fatty acids, PUFA, present in our body can readily undergo peroxidation, autoxidation, by oxygen due to the presence of double bonds. These peroxides undergo further series of reactions to generate free radicals. Examples of free radicals are Ru, Ro, O, as you have already learnt in the last unit. In fact, lipid peroxidation is a chain reaction providing a continuous supply of free radicals that initiate further peroxidation. The free radicals are extremely toxic to our tissues. The main function of vitamin E is as an antioxidant. It acts as a chain breaker and traps free radicals in cell membrane and plasma lipoproteins. It reacts with the lipid peroxide radicals formed when there is oxidation of double bonds present in PUFA. This prevents their establishing a chain reaction. The product formed between tocopherol and the free radical is relatively unreactive. It ultimately forms non-radical compounds. In this way, free radicals are prevented from causing an injury to cells. After taking part in this antioxidant reaction, vitamin E molecules get converted into the tocopheroxyl free radical product in which vitamin E is in an oxidized form. You must be aware that vitamins are present in very minute quantities in our body. This is the reason why they are called micronutrients. Hence, vitamin E molecules must be converted back into their original form, which is the reduced form. Commonly, the tocopheroxyl radical is reduced back to tocopherol by reaction with vitamin C from plasma, as we learnt earlier in Unit 9. In this way, vitamin E is regenerated to continue its antioxidant function. When vitamin C reacts with vitamin E free radical complex, the free radical is transferred to vitamin C, forming vitamin C free radical complex called monodihydroascorbate free radical. Vitamin C is also called ascorbic acid. In this process, vitamin E is regenerated in its original form. The monodihydroascorbate free radical then undergoes enzymic or non-enzymic reaction, forming ascorbic acid and dihydroascorbic acid. Neither of these compounds is a free radical and hence non-toxic. In this way, vitamin E acts as a scavenger of free radicals, protecting unsaturated fatty acids, especially membranes, from peroxidation reaction. Figure 10.5 depicts these interactions. While the presence and functions of tocopherols was known for some time, information regarding tocotrienols has been obtained in recent times. Tocotrienols occur only at very low levels in nature. In fact, it is now said that the protective effect of tocotrienols as a potent antioxidant is significantly higher than that of tocopherols. Tocotrienols help to reduce cholesterol level and have antithrombotic effects, and thus are helpful in preventing cardiovascular disease. They also demonstrate anti-cancer effects. Figure 10.5. Biological role of vitamin E. 10.3.4 Vitamin K. If you get a small cut, bleeding occurs. But very soon, without even any effort on your part, the bleeding stops. This is because a blood clot is formed. Vitamin K is required for this process. In fact, this is how this vitamin got its name, K for coagulation vitamin. Different forms of vitamin K have been described in Unit 3. Hope you are familiar with these forms. List down these forms herewith. Next, let us look at the functions of vitamin K. Functions of vitamin K. Vitamin K is required for the synthesis of various proteins which are needed for the process of blood clotting. The proteins needed for blood clotting are secreted in an inactive form called precursors. They have to be converted into the active state, following which they help in the blood clotting process. Vitamin K is required for this conversion. Hence, low levels of vitamin K result in decreased clotting of blood. The mechanism of this action has been most clearly understood in the case of one of the proteins, clotting factor, called prothrombin. Prothrombin is blood clotting factor 2. Prothrombin is synthesized as an inactive precursor called preprothrombin. This, as already mentioned, is a protein molecule, and it contains the amino acid glutamic acid units, residues, in its polypeptide chain. Conversion of preprothrombin, inactive, to prothrombin, active, requires carboxylation, introduction of COOH groups, of some of the glutamate residues. This is catalyzed by a carboxylase enzyme which requires vitamin K for its activity. The gamma carbon, fourth carbon, of glutamate residue is carboxylated to form gamma-carboxy-glutamate, as illustrated in Figure 10.6. Figure 10.6. Vitamin K-dependent carboxylation. The gamma-carboxy-glutamate residues are good chelators. What do we mean by chelators? Chelators are organic compounds which have the ability to bind to metal ions. In this case, they bind to calcium ions. The prothrombin calcium complex thus formed is converted to thrombin. Thrombin in turn converts the plasma protein fibrinogen, blood clotting factor I, to fibrin. Fibrin has a mesh-like structure and traps red and white blood cells and other plasma proteins to form the blood clot. The biological role of vitamin K as discussed above is presented in Figure 10.7. Figure 10.7. Biological role of vitamin K. Next, we shall learn about the vitamin K cycle, which you will see is the salvage pathway for vitamin K. Vitamin K cycle. Figure 10.8 depicts the carboxylation reaction and the vitamin K cycle, which is a salvage pathway for vitamin K. Vitamin K epoxide, the product of vitamin K in the glutamyl carboxylation reaction, discussed above, is recycled to vitamin K hydroquinone by enzymatic reduction. For vitamin K to take part in the carboxylation reaction, it has to be present in the hydroquinone, having two O groups form. Following carboxylation, vitamin K gets converted to the epoxide structure catalyzed by the enzyme vitamin K epoxidase. In epoxide, two carbons share an oxygen atom. Further, the two O groups get converted to double-bonded oxygen. For blood clotting to continue, the hydroquinone form must be regenerated. This happens in a two-step process. A first reduction results in loss of the epoxide oxygen, forming the quinone structure which retains the two double-bonded oxygen atoms. This reaction is catalyzed by vitamin K epoxide reductase and needs any compound having two SH sulfhydryl L groups. The two H+ atoms, one each, are added to the two carbons which have lost the epoxide oxygen. The compound now will have SS disulfide group. Vitamin K quinone undergoes the second reduction reaction catalyzed by vitamin K quinone reductase, and vitamin K hydroquinone is regenerated, as can be seen in Figure 10.8. This reduction requires the participation again of a sulfhydryl L compound to donate H+ ions for forming the O groups. In this way, a cyclic process occurs. Hence, if the blood is not well supplied with vitamin K, blood clotting will become a slow process or may not reach completion. Figure 10.8. Vitamin K cycle. Vitamin K is also needed for the synthesis of two calcium-binding proteins in the bone. These are osteocalcin and bone matrix protein. Both contain glutamate residues which have to be carboxylated to form gamma-carboxy-glutamate residues in the presence of vitamin K as discussed above. These residues have the property of chelating Ca2+ ions. This leads to deposition of calcium in the bone, i.e. bone mineralization occurs. With vitamin K, we end our discussion on fat-soluble vitamins. Let us now recapitulate what we have learned so far. Having looked at the biological role of different fat-soluble vitamins, we now move on to water-soluble vitamins. 10.4 Water-soluble vitamins. As the name suggests, these vitamins are soluble in water. Thus, they are distinctly different in property from the fat-soluble vitamins we just finished studying. Other major differences include the chemical structure and the biochemical role performed in the body. While the fat-soluble vitamins are chemically derivatives of isoprene units, as informed earlier in Unit 3 and also shown in Figure 10.9 here, there is no one common structure common to all the water-soluble vitamins. Figure 10.9. Isoprene unit fat-soluble vitamins are stored in the body, particularly the liver. Hence, accumulation of excess amounts of these vitamins, hypervitaminosis, can occur which can lead to toxic effects. However, the other group of vitamins being water-soluble is readily excreted in the urine and hence are not stored in the body. Accordingly, a state of hypervitaminosis and toxic effects are generally not seen or are rare. But at the same time, deficiency of these vitamins occurs relatively quickly on an inadequate diet. There, the metabolic stores are labile, unstable, and depletion can often occur in a matter of weeks or months. Another difference observed is the kind of biological role performed in the body. Each fat-soluble vitamin exerts a different type of effect, while a large number of water-soluble vitamins, like the so-called B-complex vitamins, have one type of activity, functioning as coenzymes in the body. Coenzymes are non-protein molecules which are required for the activity of many enzymes. You should revise the concept of coenzymes given in Unit 4. The water-soluble vitamins, which we will be discussing, may be classified into three groups depending on their function. Energy-releasing water-soluble vitamins, B1, B2, B6, niacin, pantothenic acid and biotin. Hematopoietic water-soluble vitamins, folic acid, vitamin B12, other water-soluble vitamins, ascorbic acid, vitamin C. We begin our study of water-soluble vitamins with energy-releasing water-soluble vitamins. 10.4.1 Energy-releasing water-soluble vitamins. As the name suggests, these vitamins are used in pathways which result in the production of energy. All the 6 vitamins included in this group, namely vitamins B1, B2, B6, niacin, pantothenic acid and biotin, function as coenzymes. After absorption from the intestine, each vitamin undergoes certain modifications and is converted into a form called active form which is able to take part in the reaction. Generally, these active forms are synthesized in the liver. Since these 6 vitamins have a similar function of energy generation, deficiencies of these vitamins produce a number of common and overlapping symptoms. Further, because of the central role these vitamins play in energy metabolism, deficiencies show up first in rapidly growing tissues. In many cases, the nervous tissue is also involved due to its high energy demand. In several cases, the vitamins participate in a number of chemical reactions, thus it is impossible to pinpoint the exact biochemical cause of any given symptom. You will indeed find that these facts are true if you have a look at the diseases resulting from the deficiency of these vitamins. We begin our study of this group of vitamins with thiamine or vitamin B1. 10.4.1.1 Thiamine, vitamin B1. Thiamine has a central role in energy-yielding reactions of particularly carbohydrate metabolism. The active form in which thiamine functions in the body, as you may recall reading earlier in Unit 3, is called thiamine pyrophosphate (TPP), also called thiamine diphosphate. The structure is given in Figure 10.10. Here 2 inorganic phosphate pi groups are introduced into the thiazole ring. Figure 10.10. Active form of thiamine functions of thiamine diphosphate, TDP, is the coenzyme for three multi-enzyme complexes that catalyze oxidative decarboxylation, oxidation and decarboxylation reactions. These are pyruvate dehydrogenase in carbohydrate metabolism, alpha-ketoglutarate dehydrogenase in the citric acid cycle, branched-chain keto acid dehydrogenase in metabolism of branched-chain amino acids, leucine, isoleucine and valine. You have already read about the first two reactions in Unit 6 and can refer to these sections for the details of these reactions. You have not read about the third enzyme complex. It also has similar mechanism. It converts alpha-keto branched-chain acids, obtained when branched-chain amino acids undergo transamination, to corresponding coenzyme derivatives. It is now known that enzymes of a multi-step pathway function not individually, but grouped as a complex. Further, within the protein complex is integrated the various required coenzymes. Thus, pyruvate dehydrogenase complex consists of three enzymes working sequentially: pyruvate dehydrogenase, dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase, along with five coenzymes: TDP, lipoic acid, coenzyme A, FAD and NAD+. In all these reactions, finally NAD is formed, which is oxidized in the mitochondrial electron transport chain with the formation of energy-rich ATP, as discussed in Unit 6. Additionally, TDP also functions as the coenzyme for transketolase enzyme. You have again read about this in hexose monophosphate HMP pathway. So please read that section for this role of TDP in Unit 6. 10.4.1.2 Riboflavin, vitamin B2. This vitamin, which functions as a coenzyme in energy-yielding metabolism, has two active forms: FMN, flavin mononucleotide, and FAD, flavin adenine dinucleotide. We have already studied their structures in Unit 4, Section 4.9. Enzymes which use FMN or FAD as coenzymes are called flavoproteins. What is their function? Let's find out. Functions of riboflavin. Riboflavin can function as a coenzyme because of its ability to undergo oxidation-reduction reaction, which is given in Figure 10.11. Hence, the overall reaction consists of the addition of two hydrogen atoms to the oxidized form, resulting in the formation of reduced form of flavin, FMN FMNH2, FAD FADH2. Figure 10.11. Oxidation reduction property of flavin coenzyme. After some time, all the FAD will exist in the cell as FADH2. Since FAD is a coenzyme, it is present in minute quantities and hence has to be regenerated. Hence, it is oxidized in the mitochondrial respiratory chain. This oxidation, as you already know, is coupled to phosphorylation of ADP, ultimately forming two molecules of ATP. Thus, participation of FAD in redox, oxidation reduction, reactions results in the release of utilizable energy as ATP. However, oxidation of FMNH2 does not form ATP since it is directly oxidized by oxygen or some substrate in the cell, not coupled with phosphorylation. Majority of flavoproteins contain FAD as the coenzyme. Thus, flavin coenzymes are hydrogen electron carriers in oxidation reactions. Some of the flavoproteins contain metal ions such as iron, Fe3+, and molybdenum, Mo6+, and are known as metalloflavoproteins. These metals usually participate by being alternatively reduced and oxidized, thus making the enzyme able to participate in oxidation reduction reactions. In our study of metabolic pathways, we have come across several such reactions. Given below are a few examples. FMN, L-amino acid oxidase, in kidney, which functions in oxidative deamination of naturally occurring L-amino acids. It removes the amino group as NH3 and oxidizes the remaining portion to the corresponding keto acid. You may recall reading about this reaction in Protein Metabolism, Unit 8 in subsection 8.2.2. Look up the reaction once again now. NAD dehydrogenase, metalloflavoprotein containing iron, functions in the electron transport chain. It passes H+ ions, reducing equivalents, from NAD to ubiquinone, coenzyme Q or Q. See electron transport chain in carbohydrate metabolism, Unit 6, Section 6.11. FAD, succinate dehydrogenase, functions in citric acid cycle, oxidizing succinate to fumarate. Look up the citric acid cycle in Unit 6, Section 6.5 for this reaction. The enzyme contains iron. Acyl-CoA dehydrogenase oxidizes coenzyme a ester of fatty acid at carbons 2 and 3 forming increment 2 trans-enoyl-CoA. You may recall reading about this reaction in beta-oxidation of fatty acids, Unit 7 Section 7.2. Xanthine oxidase participates in the last two steps of catabolism of the purine bases adenine and guanine, finally forming uric acid. We learnt about this in Unit 8, subsection 8.3.3. 10.4.1.3 Pyridoxine, vitamin B6. It is a generic name for 6 compounds that have vitamin B6 activity. These are pyridoxine, pyridoxal, pyridoxamine and their 5' phosphates. The structures are given in Unit 3, subsection 3.3.5 and in Unit 4, section 4.9. Look up the section now. It will help you understand the functions as discussed next better. Functions of pyridoxine. Vitamin B6 functions as a coenzyme for many enzymes involved in amino acid metabolism. There are two such reactions which require vitamin B6. These are transamination, transfer of NH2 groups from amino acids by enzymes called transaminases or aminotransferases, decarboxylation, removal of CoO group of amino acids as CO2 catalyzed by the enzymes called decarboxylases. Transamination. It is a process of combined deamination and amination according to which the amino group of one amino acid may be reversibly transferred to the keto acid of another amino acid, thus affecting amino acid-keto acid interconversion. The general reaction of transamination is given in figure 10.12. Do you recall reading about this earlier in Unit 8, subsection 8.2.1? This transamination represents a process of intermolecular, between molecules, transfer of amino groups without the splitting out of ammonia, which is highly toxic to the nervous system, neurotoxin. Figure 10.12. General reaction of transamination. The reaction is freely reversible. All amino acids except lysine, threonine, proline and hydroxyproline participate in transamination. Transaminases can function both in amino acid catabolism as well as biosynthesis. Different transaminases are known: alanine pyruvate aminotransferase or alanine transaminase and glutamate alpha-ketoglutarate aminotransferase or glutamate transaminase, present in most mammalian tissues, catalyze the transfer of amino groups to pyruvate, forming alanine, or to alpha-ketoglutarate, forming glutamate, as can be seen in figure 10.13. Serum levels of aminotransferases are elevated in some disease states. Example: myocardial infarction, viral hepatitis, etc. Figure 10.13. Action of transaminases. Approximately 60 specific reactions of amino acids involving pyridoxal phosphate have been discovered. Because B6 phosphate is involved in catabolism of amino acids, it is essential for energy production from amino acids and is considered as an energy-releasing vitamin. Decarboxylation. It is catalyzed by enzymes called decarboxylases which require B6 phosphate as coenzyme. When amino acids undergo decarboxylation, the corresponding amine is formed as shown in figure 10.14a. We have already studied about this reaction earlier in Unit 8 in subsection 8.2.7. We learned that the COOH carboxylic group is removed as CO2. This serine forms ethanolamine, figure 10.14b, while tyrosine forms tyramine, figure 10.14c. Decarboxylation of 5-hydroxytryptophan forms serotonin, 10.14d, which functions as a neurotransmitter in the body. Neurotransmitter controls transport of nerve impulse. This may explain the irritability, nervousness and depression seen with mild deficiencies and peripheral neuropathy and convulsions observed in severe deficiencies. Figure 10.14, A, B, C, D. Decarboxylation reaction. Another important enzyme which requires vitamin B6 phosphate is L-glutamate decarboxylase, converting glutamate to gamma-aminobutyrate, GABA, which is an important intermediate in the body as can be seen in figure 10.15. The enzyme occurs principally in brain tissue, where it functions as an inhibitory neurotransmitter. Figure 10.15. L-glutamate decarboxylase reaction. Pyridoxal phosphate acts as a coenzyme in other physiologically important reactions. It is required for the synthesis of delta-aminolevulinic acid, a precursor of heme. Hence, B6 deficiency can result in anemia. Pyridoxal phosphate also participates in the synthesis of the sulfur-containing amino acid cysteine. This is the reason why cysteine is a non-essential amino acid. This reaction is also important since deficiency of B6 will result in increased level of homocysteine. High levels of homocysteine, hyperhomocysteinemia, appear to be a risk factor for cardiovascular disease. Pyridoxal phosphate is a part of the enzyme glycogen phosphorylase which breaks down glycogen in the body. Hence, decreased tolerance to exercise is associated with B6 deficiency. Pyridoxal phosphate is one of the cofactors required for the conversion of the amino acid tryptophan to the coenzyme NAD+. 10.4.1.4 Niacin. It is not a vitamin in the strictest sense of the word, since some niacin can be synthesized from tryptophan. However, conversion of tryptophan to niacin is very small. 60 mg of tryptophan forming 1 mg of niacin requires other vitamins like B1, B2 and B6, and it is also very inefficient on a marginal diet. Niacin is also called nicotinic acid or sometimes vitamin B3. Besides nicotinic acid, the diet also provides us nicotinamide, which is the amide, co-NH2, of nicotinic acid. Amide is formed when amino group is substituted into carboxylic group. Nicotinamide is converted to nicotinic acid in the body. Nicotinic acid is converted into two active forms by enzymes present in the cytosol of most cells. These are NAD plus nicotinamide adenine dinucleotide and NADP plus nicotinamide adenine dinucleotide phosphate. We have already learnt about these active forms and their structures in Unit 4, Section 4.9. We suggest you open Unit 4 right away and revise the structures now, as this will help you understand the functions of niacin as a coenzyme better. Functions of niacin, NAD+ and NADP+ function as coenzymes in several oxidation reduction reactions. In Unit 4, we learned that they are coenzymes of many dehydrogenases occurring both in the cytosol as well as within the mitochondria. They are therefore key components of many metabolic pathways. Generally, NAD+ linked dehydrogenases catalyze oxidoreduction reactions in oxidative pathways, e.g. citric acid cycle, whereas NADP+ linked dehydrogenases or reductases are often found in pathways concerned with synthesis, e.g. fatty acid biosynthesis. Figure 10.16 shows the mechanism of oxidation reduction of nicotinamide coenzymes. As you can see, the nicotinamide portion takes part in this mechanism. R represents the rest of the molecule. One of the hydrogen atoms is removed from the substrate, AH2, as a hydrogen nucleus with 2 electrons forming the hydride ion H. It is transferred to position 4 of the nicotinamide ring. This results in reorganization of the double bonds in the ring. The second hydrogen removed from the substrate remains free as a hydrogen ion. Figure 10.16, Mechanism of oxidation and reduction of nicotinamide coenzyme. You must have observed that the two coenzymes are always written with plus superscript in the oxidized form. This is because the nitrogen of the nicotinamide moiety residue has 4 valencies. Normally, nitrogen has 3 valencies. Under exceptional circumstances, nitrogen can have 4 valencies when it is called quaternary nitrogen. But in such cases, the nitrogen carries a net positive charge. However, on being reduced, the nicotinamide nitrogen has only 3 valencies. Hence, NAD and NADPH are written without a plus superscript. In the section on metabolic pathways, you have come across several enzymes using NAD+ or NADP+ as the cofactor. Listed below are a few. We have also highlighted them earlier in section 4.9 in Unit 4 on enzymes. We shall not give the details of the metabolic pathway using these cofactors here since we have already discussed them earlier. You will have to go back to the appropriate sections in Units 6, 7 and 8 to get the details of these reactions. NAD+, glyceraldehyde3 phosphate dehydrogenase, look up glycolysis in Unit 6, lactate dehydrogenase, look up glycolysis in Unit 6, malate dehydrogenase, look up citric acid cycle in Unit 6, L3-hydroxyacyl-CoA dehydrogenase, look up beta oxidation in Unit 7, L-glutamate dehydrogenase, look up oxidative deamination of glutamate in Unit 8. NADP+, glucose 6-phosphate dehydrogenase, Look up pentose phosphate pathway in Unit 6, 3-ketocell reductase. Look up fatty acid biosynthesis in Unit 7, squalene synthetase. Look up cholesterol biosynthesis in Unit 7, malic enzyme. Look up fatty acid biosynthesis in Unit 7. In fact, you should make your own exhaustive list of enzymes requiring NAD+ or NADP+ as cofactor after studying the various chemical reactions of the different metabolic pathways. 10.4.1.5 Pantothenic acid. Pantothenic acid forms coenzymes involved in energy metabolism. It is absorbed readily in the intestine. A phosphate group is attached forming 4-phosphopantothene. This is followed by the addition of the amino acid cysteine. This complex is ultimately converted to coenzyme A, the active form in which pantothenic acid functions in the body. Because of the presence of cysteine, coenzyme A has a free SH group. This is the reactive part of the molecule. It is customary to abbreviate the structure as CoA, SH, as you have seen in Unit 4. Let us now study the functions of this coenzyme. Functions of pantothenic acid. The SH group can combine with carboxyl, COOH, group to form a thioester. This process of formation of CoA thioester or coenzyme A ester with the substrate is called activation of the substrate. More than 70 enzymes have been described till date that utilize CoA or its derivatives. Thus, it is not surprising that coenzyme A is required for the metabolism of fat, protein and carbohydrate. You have already come across many of these reactions, a few of which are listed below. Pyruvate dehydrogenase complex, in pyruvate oxidation in Unit 6, alpha-ketoglutarate dehydrogenase complex, in citric acid cycle in Unit 6, acyl-CoA synthetase, thiokinase, in beta oxidation of fatty acid in Unit 7. Pantothenic acid is also a component of an interesting heat-stable protein of low molecular weight called acyl carrier protein, ACP. It plays an important role in the biosynthesis of fatty acids, as you may recall reading in Unit 7, lipid metabolism. The last vitamin in this list is biotin. Let's look at its function as a cofactor in our body. 10.4.1.6 Biotin acts as a cofactor binding to its specific enzyme protein. Functions of biotin. It is intimately associated with carboxylation reactions in which carbon dioxide is added to the substrate. Hence, these reactions are called carbon dioxide fixation reactions. The enzymes are called carboxylases. There are 3 such reactions in the body. You have already come across all these 3 reactions in metabolic pathways. All biotin-catalyzed reactions also need ATP as a source of energy and magnesium ions. The enzymes which function with biotin are pyruvate carboxylase in gluconeogenesis, Unit 6, propionyl-CoA carboxylase in gluconeogenesis, Unit 6, acetyl-CoA carboxylase in fatty acid biosynthesis, Unit 7. The carboxylation takes place stepwise as given in Figure 10.17. Carbon dioxide takes part in the form of bicarbonate, hydrogen carbonate, ion as shown here with CO2 plus H2O, right pointing arrow hydrogen carbonate, left right arrow H plus HCO3. Figure 10.17. Biological role of biotin. In the first step, CO2 is added to biotin catalyzed by the enzyme biotin carboxylase forming CO2 biotin enzyme. This step is called activation of CO2. The CO2 is then transferred from biotin to the substrate, pyruvate forming oxaloacetate catalyzed by transcarboxylase, and the biotin enzyme is regenerated. Thus, the carboxylase is a multi-enzyme complex containing 3 components on one polypeptide chain, comprising a biotin carrier protein, biotin carboxylase and a transcarboxylase. Many such multi-enzyme complexes are known, e.g. beta oxidase complex and fatty acid synthase complex, about which you may recall reading earlier in Unit 7. With biotin, we end our discussion on energy-releasing water-soluble vitamins. Next group of vitamins we shall discuss now are the hematopoietic water-soluble vitamins. But first let us recall what we have learnt so far. Earlier in this section we studied about the energy-releasing water-soluble vitamins. We have seen that some water-soluble vitamins also have hematopoietic function. Let us get to know about these vitamins next. 10.4.2 Hematopoietic water-soluble vitamins. Hematopoietic water-soluble vitamins, as mentioned earlier, are those vitamins which are required for the synthesis of red blood cells in the body. Two such vitamins have been identified, which are folic acid and vitamin B12. Let's look at their metabolic role next. 10.4.2.1 Folic acid folicin, as you may already know, is the generic form of folic acid and related substances having folic acid activity. Look up the structure of folic acid in Unit 3, subsection 3.3.8. The simplest form of folic acid, as you can see, has one glutamic acid in the structure. It occurs in diet as polyglutamate derivatives with 2 to 7 glutamic acid residues. These are absorbed into the intestinal mucosal cells. Here, the extra glutamate residues are removed by conjugase, which is a lysosomal enzyme. Folic acid is then reduced by the enzyme folate reductase to dihydrofolic acid by the addition of two H+ ions to the PT-aron ring. Two more H+ ions are added to this ring by dihydrofolic reductase forming tetrahydrofolic acid. Figure 10.18 presents the synthesis of tetrahydrofolic acid. Note that these two reactions require NADPH as the cofactor. Glutamate residues are again added to tetrahydro H4 folate, and these polyglutamate derivatives are the active form. Folic acid is also stored as tetrahydrofolate polyglutamate in liver. Figure 10.18 Synthesis of tetrahydrofolate. After a basic understanding of its structure, let us look at its metabolic role in our body. Functions of folic acid H4 folate functions in what is referred to as 1-carbon metabolism. It transfers 1-carbon groups containing only 1 carbon. These include methyl, CH3, methylene, CH2, methanol, CH, formyl, CHO, and formamino, HC equals NH, groups. These groups are obtained from various compounds in the body and get attached to N5 or N10 positions forming 1-carbon derivatives which are shown in figure 10.19. All these forms are metabolically interconvertible. N5-formyl THF, also known as folinic acid, is a stable form which is used for therapeutic purposes. Figure 10.19. 1-carbon units attached to tetrahydrofolate metabolic reactions using 1-carbon derivatives are discussed below. Serine-glycine interconversion. This is a freely reversible reaction which also requires vitamin B6 phosphate. Synthesis of methionine. This reaction requires vitamin B12. Synthesis of thymidine monophosphate, TMP. TMP is a nucleotide having the pentose sugar, deoxyribose, as you have already seen in Unit 2. This reaction is of great physiological significance since TMP is a precursor of DNA synthesis, since it is a constituent of DNA. It is also required for erythrocyte formation. Hence, deficiency of folic acid causes anemia. In this reaction, dihydrofolate is formed, which is then reduced to active THF by dihydrofolate reductase. Catabolism of histidine. THF takes part in breakdown of the amino acid histidine to glutamate and is itself converted into the N5-formamino derivative. Synthesis of purine. SN10-formyl THF is the source of the carbon in position 2 of the purine ring, while N5,N10-methanol THF is the source of the carbon in position 8 of the purine ring, as we have seen in Unit 8. Thus, folic acid is required for purine biosynthesis. Purines, as you know, are the constituents of the two nucleic acids, DNA and RNA. You have already studied the structures of the intermediates involved in the above reactions. So revise them and write these folic acid reactions with structures in the space given herewith. Now, let us move on to vitamin B12. 10.4.2.2 Cyanocobalamin, vitamin B12. Vitamin B12 has been found only in animals and microorganisms and is absent in the plant kingdom. As you have already seen in Unit 3, subsection 3.3.6, it has a very big structure. The absorption of vitamin B12 requires a highly specific glycoprotein, a compound containing carbohydrate and protein, called the intrinsic factor. After absorption, the vitamin is bound by a plasma protein called transcobalamin. The vitamin is stored in the liver bound to transcobalamin. We shall look at the functions now. Functions of cyanocobalamin. Vitamin B12 functions as a coenzyme. For this, the cyanide, CN, radical is removed forming cobalamin. This is then substituted with other groups forming the coenzyme. There are two active forms, methylcobalamin. As the name suggests, a methyl group is present. It catalyzes the conversion of homocysteine to methionine. This reaction also requires THF, as can be seen in figure 10.20, deoxyadenylyl cobalamin. Here the cyanide radical is replaced by adenosine, ribose plus adenine, moiety, residue. It acts as a coenzyme for methyl-malonyl-CoA isomerase which converts L-methyl-malonyl-CoA to succinyl-CoA as shown in figure 10.20. This reaction makes it possible for the body to use propionate as a substrate for gluconeogenesis, as we studied in Unit 6. In vitamin B12 deficiency, this reaction cannot take place and THF cannot be released. Hence, all the THF is trapped as methyl THF. This is called the folate trap. It leads to impaired purine and pyrimidine synthesis, resulting in impaired DNA synthesis. This, in turn, prevents cell division and formation of the nucleus of new erythrocytes and the presence of immature erythrocytes in blood. Ultimately, there is anemia. Figure 10.20. Action of B12 coenzymes. Next, we shall look at the other water-soluble vitamins, mainly the role of vitamin C. 10.4.3. Other water-soluble vitamins. We have looked at the metabolic role of energy-releasing and hematopoietic water-soluble vitamins above. One another important water-soluble vitamins which has an important metabolic role in our body is vitamin C, which is discussed next. With this, we shall come to an end on our discussion on vitamins. So get started and get to know about the metabolic role of vitamin C. 10.4.3.1 Ascorbic acid. Vitamin C. It is necessary to have vitamin C in the diet for primates including human beings and other animals like guinea pig, bats, fishes, etc. Most other species can synthesize ascorbic acid from glucose in a multi-step pathway. Human beings do not have the enzyme for the last step and hence must have preformed vitamin in the diet. Look up the structure of ascorbic acid, which you have studied in Unit 3 subsection 3.3.9. You will notice that it possesses an endiol group on carbons 2 and 3. Endiol means two O groups between a double bonded carbon system which you have come across in reducing properties of sugars in Unit 1. Endiol group is a very strong reducing agent. Hence, the biochemical role that ascorbic acid plays is related to it being a good reducing agent. In many of these processes, ascorbic acid does not participate directly in the reaction, but is required for maintaining the metal cofactor participating in that reaction in a reduced form. This metal cofactor is necessary for the activity of the enzyme catalyzing that reaction. These enzymes are hydroxylases containing copper, Cu+, or iron, Fe2+. During hydroxylation, Cu+, cuprous ion, is oxidized to Cu2+, cupric ion, and Fe2+, ferrous ion, to Fe3+, ferric ion. Reduction back to Cu+ or Fe2+ specifically requires ascorbate, which in the process is oxidized to dehydroascorbic acid. In the body, both forms are biologically active since dehydroascorbic acid, oxidized form, can be converted back to the reduced form, ascorbic acid, by reducing agents like glutathione, a tripeptide containing the three amino acids glutamate, cysteine and glycine. Thus, vitamin C may not function like a typical coenzyme, reacting directly with the substrate. Important reactions of ascorbic acid are given herewith. Hydroxylation of basic amino acids, lysine and proline required for the synthesis of the protein collagen. Collagen is a constituent of connective tissues. Thus, ascorbic acid is important for maintenance of normal connective tissue and wound healing, since the connective tissue has to be first synthesized for a wound to heal. Vitamin C is also necessary for bone formation, since bone tissue has an organic matrix containing collagen as well as the mineral content. Collagen is a component of the ground, basic substance surrounding capillary walls and hence vitamin C deficiency is associated with capillary fragility. Synthesis of the hormones norepinephrine and epinephrine, formerly called noradrenaline and adrenaline, from tyrosine dopamine, right pointing arrow norepinephrine, right pointing arrow epinephrine. Catabolism of tyrosine where p-hydroxyphenyl pyruvate is oxidized, hydroxylated, to homogenitizic acid catalyzed by p-hydroxyphenyl pyruvate hydroxylase. Here again vitamin C helps to maintain copper in the reduced state which is required for the maximal activity of the enzyme. The next step also requires vitamin C where homogenitizic acid is oxidized to malylacetoacetate by homogentisate dioxygenase, which is a ferrous ion containing enzyme. We will find both these chemical reactions in the Unit 12, where we will discuss the inborn errors of metabolism. Bile acid formation from cholesterol requires ascorbic acid in the very first step, catalyzed by 7-alpha hydroxylase. You have already learnt about this in Unit 7. Absorption of iron is significantly enhanced by the presence of vitamin C as a dietetic student. This must be very clear to you. Dietary iron, when present in ferrous, Fe vitamins and minerals 2+ form, is more soluble and hence easily absorbed as compared to the ferric, Fe 3+ form. Vitamin C, being a reducing agent, helps to keep iron in the reduced state. Steroidogenesis, synthesis of corticosteroids in the adrenal glands. This has several steps involving hydroxylation reactions in which ascorbic acid may be required. In fact, the adrenal cortex, where the synthesis takes place, contains large amounts of vitamin C. In addition to the above reactions, ascorbic acid may act as a general water-soluble antioxidant in the body. Thus, it may act in converting the oxidized form of tocopherol, vitamin E, to the reduced form in the membrane. We have already read about this earlier in subsection 10.3.3. Besides the above functions, there is still much controversial debate going on regarding the beneficial effects of high doses, mega doses, of vitamin C in preventing the occurrence of common cold or reducing the duration of its symptoms. The first section in this unit was focused on the study of vitamins. Minerals are the other important inorganic compounds required by our body for metabolism. Next, let us get to learn about their role in body metabolism. 10.5 Minerals. An introduction. Minerals constitute a wide and complex group occurring widely in the Earth's crust. In fact, the list of minerals is continuously increasing, with new minerals being discovered. It had been realized very early in the development of nutritional science that certain minerals were essential for normal health and proper functioning of the body. The importance of minerals has been documented much before nutritional requirement for vitamins became universally accepted. Minerals are inorganic elements and hence are distinctly different in chemical nature from the other major four nutrients: carbohydrates, proteins, lipids and vitamins, which are organic compounds. Another major difference is the type of function performed. Each mineral in the body has an exclusive function. On the other hand, each of the other four nutrients has generally at least one common function in the body. Carbohydrates, best source of energy; proteins, structural component; fats, high energy fuel; vitamins, cofactors. Although in respect to their amounts, the mineral elements are relatively minor components of the tissues, they are essential to many vital processes, such as provide a suitable medium for protoplasmic activity. Many salts are important in acid-base equilibria and the osmotic control of water balance, and certain tissues like bones and teeth have a high mineral content which accounts for their hardness and rigidity. The metabolism of food minerals does not involve the radical changes of molecular form that are found in carbohydrate, protein and lipid metabolism. You have already learnt in detail the extensive metabolic reactions undergone by the above three classes of nutrients in the body. The positive mineral ions such as calcium, magnesium, potassium and sodium taken in our food as salts of organic or inorganic acids or associated with proteins or lipids after absorption are associated with just such negative ions in the body. For example, the calcium ion may partly become associated with plasma protein or protoplasmic protein or organic or other inorganic acids. The phosphate radical can be converted into any of a great number of organic esters in the blood or tissue cells. The positive and negative mineral ions, not used as body structural units, undergo in general no greater chemical alteration than exchange of partners during metabolism and excretion. Most minerals, except sodium and potassium, form salts and other compounds that are relatively insoluble. Hence, they are not readily absorbed and most ingested minerals are excreted in feces. Mineral absorption often requires specific carrier proteins. These proteins can chelate, combine, with minerals and carry them into the intestinal epithelial cell. In fact, the synthesis of these proteins serves as an important mechanism for control of mineral levels in the body. Even transport in the body and storage in the tissues require specific binding proteins. Excretion of most minerals is accomplished by the kidneys. Many minerals are also secreted into the digestive juices and bile and subsequently lost in the feces. You may be already familiar with the classification of minerals. A simple classification is also presented herewith for your reference. Classification of minerals. One commonly used system of classification is based on the amount of mineral required, present in the body. Accordingly, there are two major classes: principal or macrominerals, macroelements; trace or microminerals, microelements. Minerals may also be classified on the basis of their functions. In this unit, we are only looking at the biological functions. It would be very meaningful to have an overview of the functions. This is given in Table 10.1. You may have already learnt about these functions in the Advanced Nutrition course in Units 9 and 10. Here, we shall look at these functions from the biochemical point of view. Table 10.1. Classification of minerals according to their function. Function: minerals, structural function, calcium, phosphorus, magnesium involved in membrane function, principal cations of extracellular and intracellular fluids, respectively, sodium, potassium function as prosthetic groups in enzymes, iron, zinc, copper, cobalt, selenium, molybdenum regulatory role or role in hormone function, calcium, chromium, iodine, magnesium, manganese, sodium, potassium may occur in foods and known to be toxic in excess, aluminium, arsenic, antimony, fluoride, lead, mercury etc. We will now go on to the specific functions of each. We start with macrominerals. 10.5.1 Macrominerals. As the name suggests, these minerals are required in comparatively larger amounts in the diet, generally greater than 100 mg per day. It follows that they are also present in greater amounts in the body. There are 7 essential principal elements: calcium, magnesium, sodium, potassium, phosphorus, sulfur and chlorine. They constitute 60 to 80% of all the inorganic material in the body. Let us get to know about them. 10.5.1.1 Calcium. Calcium is present in the body in larger amounts than any other cation, a positively charged particle, as much as 1200 grams in a 70 kilograms adult. Almost all of it, about 99%, is in bones and teeth. The other 1% is in blood, lymph and soft tissues. Most skeletal calcium is deposited in the form of a crystalline complex called hydroxyapatite, calcium phosphate 6O2. Bone also contains considerable amounts of non-crystalline calcium phosphates and carbonates, as well as small amounts of other salts. These minerals comprise about 50% of the total skeletal muscle. The remaining mass consists of an organic matrix of proteins, glycoproteins and proteoglycans on which the calcium salts are deposited. Glycoproteins and proteoglycans are proteins combined with different types and proportions of carbohydrates. Even though bones and teeth are rigid structures, they continuously undergo remodeling in which calcium and phosphorus are removed each day and replaced by new calcium and phosphorus molecules. Let us learn about the functions of calcium in greater details. Functions of calcium. In higher mammals, the most obvious role of calcium is structural or mechanical. Calcium is intimately related with bone development and teeth formation. The organic matter is first formed by the bone cells and then deposition of bone mineral takes place in the form of mainly calcium phosphate. In the tooth, enamel is the most highly calcified part. Without bone development, there can't be growth, so calcium is intimately related with and essential for growth. Blood clotting process requires calcium ions for conversion of prothrombin to thrombin. This involves conversion of glutamate residues to gamma-carboxy-glutamyl residues, GLU right pointing arrow GLA. These residues serve as high-affinity binding sites for Ca2+ and hence can chelate calcium ions. Thrombin then acts on fibrinogen to form fibrin. This forms a mesh and entangles the blood cells to ultimately form the blood clot. Besides prothrombin, also called factor II, several other proteins of the blood clotting system, factors 7, I, X and X and proteins C and S, are activated by calcium ions. Each contains between 4 and 6 gamma-carboxy-glutamyl residues which chelate calcium ions and so permit the binding of the blood clotting proteins to membranes. We have already learnt about the formation of gamma-carboxy-glutamate residues in functions of vitamin K in this unit in subsection 10.3.4. Milk clotting also requires Ca2+. This is important in the digestion of milk in infants. Calcium ion is directly related to muscle contraction and nerve impulse transmission. In the absence of calcium, all types of muscles lose their ability to contract. Membrane permeability is decreased by calcium and capillary permeability is increased by calcium. Calcium is a mediator of hormone action. Hormone carries message to the cell to carry out certain chemical reactions. Hence, hormone is called the first messenger. On reaching the cell, the communication of this message from the hormone to the intracellular compartment requires intermediary molecules. And in many cases, calcium functions as this molecule. Hence, calcium is called the second messenger. You will read more about this in the next unit on hormones. Several enzymes and proteins are regulated by calcium. A few examples are given below. Adenylyl cyclase, CA2+ dependent protein kinases, phospholipase A2, phosphorylase kinase, calbindin, calsequestrin, calmodulin, troponin C. You have come across these enzymes in metabolic pathways. It would be a good idea to locate them in the appropriate section. Calbindin is a protein which binds with calcium and helps in the intestinal absorption of calcium. Calsequestrin is again a calcium-binding protein which helps in calcium storage. Activation of many enzymes by calcium is mediated by calmodulin. Calmodulin has 4 calcium-binding sites and full occupancy of these sites leads to marked conformational, structural changes which allow calmodulin to activate enzymes. Troponin C is a muscle protein. Let us now look at the mechanism of action of calcium. Mechanism of action of calcium. The mechanism of action of calcium is linked to its ability to bind with a large number of cell proteins. Let us see how. The calcium ion, Ca2+, is able to form coordination bonds with up to 12 oxygen atoms. This makes calcium nearly unique among all cations in its ability to fit neatly into the folds of the peptide chain. By binding with oxygen atoms of glutamic and aspartic acid residues, calcium stiffens the protein molecule and fixes its tertiary structure. You may recall studying about the tertiary structure of proteins in Unit 2. Binding of calcium to a large number of cell proteins results in the activation of their unique functions. These proteins range from those involved with the cell movement and muscle contraction to nerve transmission, glandular secretion, and even cell division. In most of these situations, calcium acts as both a signal transmitter from the outside of the cell to the inside and an activator of the functional proteins involved. In fact, ionized calcium is the most common signal transmitter in all biology. We will learn about the messenger function of calcium later in the next unit. When a cell is activated, e.g. a muscle fiber receives a nerve stimulus to contract, the first thing that happens is that calcium channels in the plasma membrane open to admit a few calcium ions into the cytosol. We learnt about this mechanism, if you recall, in the Applied Physiology course in Unit 9. These bind immediately to a wide array of intracellular activator proteins, which in turn release a flood of calcium from the intracellular storage vesicles. This second step quickly raises cytosol calcium concentration and leads to activation of the contraction complex. 2 of the many calcium-binding proteins are of particular interest here. A. Troponin C. After it has bound, calcium initiates a series of steps that lead to the actual muscle contraction. And B. Calmodulin, a second and widely distributed calcium-binding protein, activates the enzymes that break down glycogen to release energy for contraction. This involves the enzyme phosphorylase kinase. This enzyme has 4 subunits alpha, beta, gamma and delta. The gamma subunit binds 4 calcium ions and is identical to calmodulin. The binding of Ca2+ activates the catalytic site of the gamma subunit, resulting in the activation of the enzyme glycogen phosphorylase, which causes the breakdown of muscle glycogen, glycogenolysis. Thus, activation of muscle contraction and glycogenolysis are carried out by the same Ca2+ binding protein, ensuring their synchronization. When the cell has completed its assigned task, the various pumps quickly lower the cytosol calcium concentration and the cell returns to a resting state. Next, we move on to phosphorus. 10.5.1.2 Phosphorus exists in cells as a free ion at a concentration of a few milliequivalents per liter. It is also an integral component of many physiologically important molecules including nucleic acids, nucleotides, phospholipids and some proteins. In the extracellular space, phosphate circulates as a free ion. It is present with calcium as hydroxyapatite, a major component of bone. All cells possess enzymes, kinases, that can attach phosphate to other molecules, as well as enzymes, phosphatases, that can remove phosphate from molecules, as you may have seen in our study so far. A normal 70 kilograms man contains about 700 grams of phosphorus. Phosphorus as phosphate is found in every cell of the body, but most of it, about 80%, is combined with calcium in bones and teeth. About 10% is in combination with proteins, lipids and carbohydrates and with other compounds in blood and muscle. The remaining 10% is distributed in various chemical compounds in the body. Phosphate is the most abundant anion in the cell. There is a close association between calcium and phosphorus in the body, as you may recall reading earlier in subsection 10.3.2 under vitamin D functions. Let us look at the functions of phosphorus next. Functions of phosphorus. Phosphorus is involved with formation of bone and dental tissue. Participation in the structure of all body cells as phospholipids, which are the constituents of plasma cell membranes. Participation in reactions resulting in the liberation of energy. Various high-energy molecules like ATP, GTP, creatine phosphate contain phosphorus. When the phosphate bond is broken, energy is released, which is used for carrying out chemical reactions in the body. Hence, high-energy phosphates, such as ATP etc., are referred to as energy currency of the cell, as highlighted earlier too in Unit 8. Influencing acid-base balance of blood. Phosphorus is present in compounds like disodium hydrogen phosphate, sodium hydrogen phosphate, and sodium dihydrogen phosphate, sodium hydride PO4, which act as buffers and maintain pH of blood within the desirable limits. Constituent of various physiologically important molecules, nucleic acids, DNA and RNA. These compounds, as you may already know, are important from the standpoint of genetics and protein synthesis. You have already learnt about these compounds in Unit 2, under Section 2.8 Structure of Nucleic Acids. A variety of coenzymes NAD+, NADP+, FMN, FAD. You are already aware of a large number of reactions in various metabolic pathways which require these coenzymes for the enzymes to become functional. Regulation of metabolic pathways. It is very essential to have mechanisms for regulating all the metabolic pathways. One such mechanism is by covalent modulation. This involves phosphorylation-dephosphorylation of enzymes. While certain enzymes become activated in the phosphorylated form, others are active in the dephosphorylated form. A number of hormones depend upon phosphorylation for their activation. So now you realize how important this compound is. Next, let us learn about magnesium. 10.5.1.3 Magnesium. Occurrence of magnesium is widespread in both plant and animal tissues. The total body concentration of magnesium in a healthy adult is about 20 to 28 grams. Somewhat more than half, 53%, of the total body magnesium is in bone as magnesium phosphate Mg3, PO4, 2, and almost all the rest is in soft tissue. Magnesium is the most abundant divalent mineral cation in cells and is second only in electrolyte quantity to monovalent potassium. Most intracellular magnesium exists in bound form. Let us look at its functions next. Functions of magnesium. Being part of the bone tissue, magnesium has an important structural function. Magnesium is involved in more than 300 essential metabolic reactions. Magnesium ion, Mg2+, forms complexes with a variety of organic molecules having biological activities. The binding of functional groups is in descending order: phosphate greater than carboxylate greater than hydroxyl, in terms of both relative importance and binding affinities. Mg2+ is essential for many enzymatic reactions and has two general interactions as highlighted here with A. Mg2+ binds to the substrate, thereby forming a complex with which the enzyme interacts, as in the reaction of the kinase enzymes with Mg-ATP and B. Mg2+ binds directly to the enzyme and alters its structure and/or serves a catalytic role, as in the case of enzymes like exonuclease, topoisomerase and ANA and DNA polymerases. Thus, magnesium plays an important role in glycolysis, the citric acid cycle, gluconeogenesis, lipid metabolism, amino acid metabolism and nucleic acid metabolism. The transketolase reaction involving thiamine and the transfer of CO2 to biotin in carboxylation reactions requires Mg2+. Glutathione, a key intracellular antioxidant, has Mg2+ requirement for its synthesis. Magnesium is important in energy metabolism since ATP, the free energy currency for all cellular processes, exists in all cells primarily as Mg-ATP. Magnesium, calcium and some other cations react with hydrophilic polyanionic carboxylates and phosphates of the various membrane components to stabilize the membrane and thereby affect fluidity and permeability. Cyclic AMP, CAMP, which acts as a second messenger in hormone action, is formed from Mg-ATP and the enzyme adenylyl cyclase, which is activated by magnesium through its two binding sites. Hence, magnesium is required for hormone action. Before we move any further, let us assess what we have learned so far by answering the questions given in Check Your Progress exercise 4. We move on to the next group of minerals classified as microminerals. 10.5.2 Microminerals. Microminerals occur in living tissues in minute amounts. In fact, early workers who were unable to measure their precise concentrations with the methods then available frequently referred to them as occurring in traces. For this reason, they came to be known as trace elements. Other popular names used include minor elements or oligoelements, from the Greek oligos, meaning scanty. The microminerals are required in amounts less than 100 mg per day. The trace elements may be subdivided into three groups: essential trace elements, iron, copper, iodine, zinc, manganese, cobalt, molybdenum, selenium, chromium. These have been shown to be dietary essentials vital to the enzymic processes of the living cell. Possibly essential trace elements: nickel, tin, vanadium, cadmium, silicon, barium, strontium. They exhibit some metabolic activity, revealed by both in vivo and in vitro studies. Non-essential trace elements: aluminium, boron, lead, mercury, fluorine, arsenic. By far, the greatest numbers are apparently inert in the sense that they have not been shown to perform any vital function or to affect living processes in the concentrations in which they normally occur. However, there is no permanency to membership, particularly in the second and third groups. When the concept of trace elements was elucidated, only iron and iodine were classified as dietary essentials. As physiological and analytical techniques improved, distinctive functions of various mineral elements were identified and these were included in the first group. This is an ongoing process. Further, it is pertinent to mention here that a reverse thought process has occurred in India with respect to fluorine. This element has long been considered as essential for development of teeth. It still is in the Western countries. However, extensive research in India has shown that fluorine occurs in extremely high concentration in our soil and water. Accordingly, it may be consumed in large amounts, leading to toxicity conditions including excessive mineralization in bones and teeth, leading to irreversible crippling and loss of teeth. Additionally, excessive fluoride can cause extensive damage to epithelial lining of tissues. Hence, fluorine definitely belongs to the non-essential group. The 20 to 30 trace elements of this group that occur in living tissues, a considerable number, notably aluminum, silver, lead, gold, bismuth, are believed to be acquired and accumulated as environmental contaminants, and their presence merely reflects the contact of the organism with its environment. We begin our study on each of these microminerals with iron. 10.5.2.1 Iron. Iron was a familiar metal in most of the ancient civilization of the Mediterranean coast and hence led to its early medicinal use. In the earliest manuscript of Egypt, rust was prescribed as an ointment to prevent baldness. Iron is one of the most abundant elements in the Earth's crust. However, the body of an adult weighing 70 kilograms contains only 4 to 5 grams of iron, i.e. 0.006 to 0.007% of total body weight. Hence, it is classified as a trace element. Most of the body iron exists in complex forms bound to protein, i.e. as heme compounds, porphyrin plus iron, notably hemoglobin, in blood, and myoglobin, in muscle, or as non-heme protein-bound compounds such as ferritin, storage form of iron, and transferrin, transport form of iron. Additionally, it is also present in various enzymes either as heme or as non-heme iron. However, this constitutes less than 1% of the total body iron. We are all familiar with the functions of iron. Let us refresh our knowledge regarding this important mineral. Functions of iron. The major use of iron is for oxygen transport by hemoglobin. The importance of this function cannot be overstressed since oxygen is central to respiration. The process of respiration essentially involves oxidative reactions. Hence, oxygen must be supplied to all the tissues. When oxygen enters the lungs, it combines with the iron-containing protein, hemoglobin, present in RBCs forming the complex oxyhemoglobin. On reaching the tissues, the complex dissociates releasing oxygen. Simultaneously, CO2 formed as a waste product in catabolic reactions in the tissues, enters RBC and combines with hemoglobin to form hemoglobin carbamate. On reaching lungs, hemoglobin carbamate dissociates, releasing CO2, which is exhaled. Oxygen requirement of muscle cells is high because of high level of metabolic activity. To ensure availability, oxygen is stored combined with iron-containing muscle protein, myoglobin. When strenuous exercise markedly lowers the oxygen content of muscle cells, myoglobin releases oxygen for mitochondrial synthesis of ATP, permitting continued muscular activity. This could well be the reason for fatigue experienced in iron deficiency condition of anemia. Insufficient iron results in the decreased synthesis of hemoglobin and myoglobin with consequent effects. Iron, as a part of heme, is a constituent of enzymes peroxidase and catalase which catalyze oxidation-reduction reactions. Physiologically, these two enzymes are very important since they bring about degradation of toxic peroxide molecules as presented herewith. H2O2 plus AH2, right pointing arrow 2H2O plus A2H2O2, right pointing arrow 2H2O plus O2. Accumulation of peroxides can lead to generation of free radicals, ROO, RO, about which you learnt in the last unit, which in turn can disrupt membranes and could cause cancer and atherosclerosis. Iron, as a part of heme, is present in various cytochromes. Cytochromes are iron-containing heme proteins. Cytochromes are components of the mitochondrial electron transport chain B, C1, C, A and A3, cytochrome oxidase. Here they function as carriers of electrons from flavor proteins on the one hand to cytochrome oxidase on the other. Cytochromes are also found in other locations, e.g. the endoplasmic reticulum contains cytochromes P450 and B5. In liver microsomes, these two molecules have an important role in detoxification, converting toxic compounds into non-toxic intermediates. Iron is also present as iron-sulfur combination, FeS, in non-heme enzymes like flavor proteins, metalloflavor and with cytochrome B. The sulfur and iron are thought to take part in the oxidoreduction mechanism, with the iron atom undergoing oxidoreduction between Fe2+ and Fe3+. Succinic dehydrogenase, an enzyme of the citric acid cycle, contains Fe(S) and oxidizes succinate to fumarate. Another metalloflavor enzyme is NAD dehydrogenase containing FeS and FMN. It oxidizes the reduced NAD of the respiratory chain and passes reducing equivalents to ubiquinone or coenzyme Q, or also simply called Q. The enzyme aconitase, which functions in the citric acid cycle, also contains iron-sulfur cluster at its active site. It intimately links the iron content of cells with energy production through oxidative phosphorylation, both in carbohydrate and lipid metabolism. When there is sufficient iron in mitochondria or in cytosol, aconitase will contain 4 atoms of iron and 4 atoms of sulfur. This is the enzymatically active form of aconitase. However, in iron deficiency state, the iron-sulfur complex is modified with only 3 atoms of iron. In this form, aconitase is enzymatically inactive, but becomes the iron regulator protein, IRP. It inhibits synthesis of apoferritin, iron storage protein, but stimulates the synthesis of delta-aminolevulinic acid synthase, enzyme involved in synthesis of heme, and transferrin, iron transport protein, receptor. In this way, iron uptake and heme synthesis are regulated at the cellular level to meet the needs of oxidative phosphorylation via the citric acid cycle. Going through the discussion above, we realize what important biological role iron has in the body. Though required in small amounts, it performs a few major functions. Let us next study about iodine. 10.5.2.2 Iodine. You are all aware that it is important to use iodized salt, i.e. salt to which the microelement iodine is added. In 1997, Parliament passed law making sale of iodized salt mandatory. In fact, extensive advertisements were carried in print and audiovisual media to educate the common person about the need to use iodized salt. Like iron, iodine too has had a long and fascinating history in human medicine. The ancient Greeks are reputed to have used burnt sponges successfully in the treatment of human goiter. Iodine is present in abundance in sponges. Use of salts of iodine for the treatment of goiter is documented as early as 1820. The element was discovered in the thyroid gland in 1895. It is clear that insufficient quantities of iodine in the diet results in the disease goiter. Goiter, as we all know, is characterized by an enlarged thyroid gland, which becomes visible in the region of the neck. Goiter occurs when soil and water have low levels of iodine and consequently the food, plant and animal sources, and water we consume are not able to meet our daily requirement. Hence, goiter will become prevalent over a geographical area and this is referred to as endemic goiter. In our country too, endemic goiter has been an exceedingly serious public health problem. This is associated with cretinism, feeble-mindedness and general physical and mental degeneration. Cretinism is a condition originating in fetal life or early infancy due to severe thyroid deficiency, characterized by stunting of physical and mental development. Since iodine is a micronutrient, the very small amounts required can easily be met by using iodized salt. Surprisingly, recently the government has not made sale of iodized salt mandatory, leaving the choice of selection to the consumers. The healthy human adult body contains 15 to 20 milligrams of iodine, of which about 70 to 80% is in the thyroid gland. The thyroid gland, which weighs only 15 to 25 grams, possesses a remarkable power for concentrating, accumulating iodine. The amount of iodine in the gland is closely related to the iodine intake. The content may be reduced to 1 milligram or less in the iodine-deficient enlarged thyroid. The enlargement of thyroid, hyperplasia, then occurs as a compensatory mechanism to utilize as efficiently as possible the decreased amounts of iodine. What is the role of iodine, which makes it so essential? Let us find out. Functions of iodine. Iodine is an integral part of the hormone secreted by the thyroid gland. In fact, there are two compounds secreted by the thyroid gland which are physiologically active: iodothyronine, T3, and tetraiodothyronine, T4 or thyroxine. Functions of iodine are essentially the functions of the thyroid hormone, which will now be discussed in detail. All cells of the body, with the possible exception of adult brain and testes, are target cells for thyroid hormone. Thyroid hormones bind to specific high-affinity receptors in the target cell nucleus. T3 binds with approximately 10 times the affinity of T4. The role of thyroid hormone include thermogenesis and oxygen consumption. Increased heat production and oxygen consumption are characteristics for most tissues responding to thyroid hormone, brain, testes and spleen excluded. Thus, thyroid hormone activity is intimately related to the basal metabolic rate, BMR. Much of the energy utilized by a cell is for driving the Na plus, K plus ATP pump. Thyroid hormones enhance the function of this pump by increasing the number of pump units. Since all cells have the pump and virtually all cells respond to thyroid hormones, this increased utilization of ATP and the associated increase of oxygen consumption via oxidative phosphorylation could be the basic mechanism of thyroid hormone function. Obesity in some patients has been suggested to be the result of decreased energy and heat production due to diminished ATPase activity. Metabolic effects of T3 and T4. These include alterations in metabolism of carbohydrates, proteins, lipids, electrolytes and water. Thyroid hormone effects on carbohydrate metabolism involve increased intestinal absorption of glucose balanced by increased glucose utilization. The net effect is one of hyperglycemia and an abnormal glucose tolerance curve. Thyroid hormones enhance general protein synthesis and cause a positive nitrogen balance. Thyroid hormones induce or repress proteins by increasing or decreasing gene transcription mechanism. Thyroid hormones act in conjunction with pituitary growth hormone, GH, as the principal anabolic agents during growth and in maintaining protein stores. T3 enhances transcription of the GH gene so that more GH is produced. Synergistic effects of the two can be demonstrated on protein synthesis in the liver. Very high concentrations of T3 inhibit protein synthesis and cause negative nitrogen balance. Cholesterol blood levels are high in hypothyroidism, and the high levels can be decreased with thyroid hormone administration. There is an increased lipid utilization with thyroid hormone. Retention of water and electrolytes in the hypothyroid state can be reversed by thyroid hormone administration. Altering the thyroid hormone state in the human causes well-known changes in the central nervous system, in nerve and muscle function, in the gastrointestinal tract, and in the vascular system. The skin is a good indicator of the thyroid state. In hyperthyroidism, the skin is smooth, warm and moist as a result of vasodilation. In contrast, the skin is cold and has a rough texture due to vasoconstriction in the hypothyroid state. The characteristic accumulation of fluid and mucopolysaccharides with the resulting puffiness, pitting edema, of the skin gives rise to the adult hypothyroid state called myxedema. The heart reflects the changes in thyroid state, having a slow rate and decreased blood flow in the hypothyroid condition. Hyperthyroid state has an effect on the heart with increased heart rate and cardiac hypertrophy. Thyroid hormones are known to be important modulators of developmental processes. The important role of thyroid hormone in human development is apparent in cretinism, a condition brought about by thyroid deficiency during the prenatal period, resulting in serious detriment in both mental and physical development in the growing child. 10.5.2.3 Zinc. Evidence of essentiality of zinc was demonstrated in plants in 1869 and in animals in 1934. Zinc is the most abundant intracellular trace element. It has been estimated that the newborn contains approximately 60 mg zinc. During growth and maturation, the zinc concentration of the human body increases to approximately 3 OMIL/LICENT/IGRAM/PERGRAM. The adult total body zinc content ranges from about 1.5 grams in women to 2.5 grams in men. Zinc is present in all organs, tissues, fluids and secretions of the body. Zinc is primarily an intracellular cation, with well over 95% of total body zinc found within the cells. Zinc is associated with all organelles of the cell, but about 60 to 80% of the cellular zinc is found in the cytosol. Among the major organs and tissues in a normal adult man, skeletal muscle and bone contain approximately 57% and 29% respectively. Skin and liver contain 6% and 5% zinc respectively. While brain contains 1.5% of total body zinc, kidneys, heart, hair and blood plasma contain minute quantities, 4% of total body zinc. Cornea is the tissue with the highest zinc concentration in the body. In biological systems, zinc is virtually always in the divalent state, zinc 2+. Zinc readily complexes to amino acids, peptides, proteins and nucleotides. Zinc has an affinity for thiol, SH, and hydroxy, O, groups and for ligands containing electron-rich nitrogen as a donor. Zinc does not exhibit any direct redox chemistry, i.e. loss and gain of electrons. Let us look at the role of zinc. Functions of zinc. Zinc is an important trace element required for normal maintenance of human health. It is involved in a multitude of diverse catalytic, structural and regulatory functions. Over 300 zinc metalloenzymes have been described to date, including carbonic anhydrase, phosphatases, alcohol dehydrogenase, glutamate dehydrogenase, both DNA and RNA polymerases, fructose bisphosphatase etc. A critical function of zinc is its role in the structure and function of biomembranes. Loss of zinc from the membrane results in increased susceptibility to oxidative damage, structural strains and alterations in specific receptor sites and transport systems. Zinc plays an important role in cell multiplication and cell growth. Hence, reduced cell replication is an early event in zinc deficiency. This has been related in part to the role of zinc in protein and nucleic acid synthesis. Zinc deficiencies in children are usually marked by poor growth and impairment of sexual development. Mild zinc deficiency may also affect the quality of growth. In both children and adults, zinc deficiencies result in poor wound healing. Zinc in many proteins is present in what is called the zinc finger motif. Zinc fingers enable polypeptides that are too small to fold by themselves to fold stably when stabilized by bound zinc. Several transcription factors have been reported to contain zinc finger regions. Zinc also serves as a stimulator of trans-acting factors responsible for regulating gene expression. This function has been studied most extensively for the expression of the protein metallothionein, MT, which is a copper-binding protein. High intake of zinc induces synthesis of MT in the mucosal cell. This protein sequesters, holds, copper, making it unavailable for serosal transfer and thus decreases copper absorption. Some of the effects of zinc deficiency in humans appear to be mediated through effects on hormonal function. Hormones reported to be affected by zinc status in humans include growth hormone, the gonadotropins and sex hormones, prolactin, thyroid hormones, corticosteroids and insulin. Zinc plays a role in stabilizing hormone receptor complexes. We will learn about hormone receptor complexes in the next unit. High levels of oxidation in the tissues lead to the generation of free radicals. These free radicals are implicated in the onset of many degenerative diseases like cancers and diabetes. Adequate zinc intake along with other antioxidants helps in reducing the risk of acquiring these diseases. Further low zinc levels in the body leads to immune deficiencies and susceptibility to a host of infections and non-communicable diseases. Zinc is required for cytokine production by monocytes and T cells. Moderate zinc deficiency leads to growth retardation, rough skin and hypergonadism in males. During the 1960s, in the Middle East countries like Iran and Egypt, young boys exhibited severe growth retardation and were shorter in structure. Sex organs and secondary sexual characters were not developed. Additionally, they suffered from anemia. They had lower zinc levels and were termed zinc dwarfs of Middle East. On supplementation with zinc, the symptoms disappeared. This emphasizes the role of zinc in growth and normal development. Zinc deficiency affects women too and may cause dwarfism and amenorrhea. Zinc is also present in guston, a salivary polypeptide that appears to be necessary for normal development of taste buds. This zinc deficiency also leads to decreased taste acuity, hypogeusia. 10.5.2.4 Selenium. Interest in the biological significance of selenium was for many years confined to its toxic effects upon animals. In fact, alkali disease in cattle living in soils with high selenium content was known to early settlers in North America. Human beings in these areas can also suffer from selenium poisoning. Condition of excessive selenium is also referred to as selenosis. The first demonstration of a biochemical function of selenium in animals came in 1973, when it was shown to be a constituent of the enzyme glutathione peroxidase. The importance of selenium in human nutrition was shown in 1979 when Chinese scientists reported that selenium supplementation prevented development of a cardiomyopathy, moderate to severe heart enlargement with varying degrees of heart insufficiency, known as Cation disease, in children living in low selenium areas. It is now well known that selenium is an essential element. Why? Read the functions of selenium next and find out. Functions of selenium. Most of the selenium in biological systems is present in amino acids as constituents of proteins. 11 selenoproteins have been characterized. Cysteine is a sulfur-containing amino acid. However, the sulfur atom can be replaced by selenium and the compound is called selenocysteine. While not normally considered an amino acid present in proteins, selenocysteine occurs at the active site of several enzymes. Examples include glutathione peroxidase, thioredoxin reductase, and iodothyronine deiodinase. You have already learned the importance and functioning of glutathione peroxidase in pentose phosphate pathway in Unit 6. This enzyme converts toxic hydrogen peroxide, hydrogen oxide, to non-toxic water in the presence of reduced glutathione. Thus, selenium functions as a scavenger of peroxides. This reaction is important since accumulation of H2O the 2nd of may decrease the lifespan of the erythrocytes by causing oxidative damage to the cell membrane, leading to hemolysis. In fact, it is now recognized that importance of selenium is on par with vitamin E in maintaining desirable redox potential in the cell. The sulfur atom of methionine can also be replaced by selenium forming selenomethionine, which has the same functions as normal methionine. 10.5.2.5 Copper has been used therapeutically since at least 400 BC when Hippocrates prescribed copper compounds for pulmonary and other diseases. The presence of copper in plant and animal tissues was recognized almost 150 years ago. The first conclusive evidence that copper is an essential dietary component emerged from various studies which indicated that copper in addition to iron was necessary for hemoglobin formation in the rat. The demonstration of the role of copper in hematopoiesis stimulated interest in the biological function of copper at the cellular level. It was found that certain naturally occurring diseases in grazing sheep and cattle were found to be caused by dietary deficiency of copper or to respond to copper therapy. Copper deficiency was shown to be a causal factor in a disease of sheep and cattle characterized by diarrhea, anorexia, and anemia. Copper was shown to be vitally concerned in the process of pigmentation, keratinization of wool, bone formation, reproduction, and myelination of the spinal cord in addition to that of hematopoiesis. The healthy human adult body has about 50 to 120 milligrams of total copper, located mostly in bone, liver, kidney, and muscle. This amount is very little when compared with other trace elements such as iron and zinc. Copper present in plasma is transported bound to a protein called ceruloplasmin. It has a pale blue color because of its high copper content and carries 90% of the copper present in plasma. Each molecule of ceruloplasmin binds 6 atoms of copper very tightly. Albumin carries the other 10% of the plasma copper. The functions of copper include functions of copper. Copper functions as a part of a number of proteins including many important enzymes. Some of these are copper-binding proteins, metallothionein, albumin, blood clotting factor V, amine oxidases, peroxidases, cytochrome C oxidase, superoxide dismutase, tyrosinase, C18, increment 9 desaturase. Several important amine oxidases are cuproproteins. Relatively small amounts of these enzymes are found circulating in blood plasma where they inactivate and catabolize physiologically active amines such as histamine, tyramine, and polyamines. Histamine stimulates acid secretion in the stomach. In allergic reactions throughout the body, histamine is released in response to exposure to antigens. Lysyl oxidase is a unique amine oxidase and it acts on lysine residues of collagen and elastin and deaminates them to form allicin, which is needed for cross-links. This enzyme functions in the formation of connective tissues including bone, blood vessels, skin, lungs, and teeth. Peroxidases. Ceruloplasmin is also called peroxidase I. It contains 6, possibly 7, atoms of copper per molecule. It catalyzes the oxidation of ferrous iron and plays a role in the transfer of iron from storage to sites of hemoglobin synthesis. Ceruloplasmin also oxidizes aromatic amines and phenols. Peroxidase 2 also catalyzes the oxidation of ferrous iron and accounts for about 5% of the peroxidase activity in human plasma. Cytochrome C oxidase. It contains 2 or 3 atoms of copper per molecule. It is present in mitochondria of cells throughout the body and is the terminal link in the electron transport chain. The activity of this enzyme is highest in heart and high in brain, liver, and kidney tissues. You can revise this portion in Unit 6. Superoxide dismutase. It is present in high amounts in the lungs, thyroid, and uterus and in small amounts in blood plasma. It functions as a scavenger of superoxide radicals and protects against oxidative damage. Tyrosinase. It catalyzes the conversion of tyrosine to dopamine and the oxidation of dopamine to dopaquinone, which are steps involved in the synthesis of body pigment melanin. Melanin is responsible for the color in hair, skin, and eyes. Deficiency of tyrosinase in skin leads to albinism. Dopamine is also a neurotransmitter, controlling nervous activity. C18, increment 9 desaturase. This enzyme is responsible for converting stearic acid, C18 saturated fatty acid, to oleic acid, C18 monounsaturated fatty acid. This may account for the fact that dietary stearic acid does not have the cholesterol-raising property of other saturated fatty acids. Metallothionein, MT. MTs are the small non-enzymatic proteins rich in cysteine that are responsible for binding copper. Each molecule can bind 11 or 12 copper atoms, as well as zinc and cadmium. They appear to play a role in metal storage and sequester excess metal ions, preventing toxicity. The concentration is highest in the liver with small amounts in the blood plasma. Albumin. It is the most prevalent protein in blood plasma and interstitial fluids. Albumin binds and transports copper and also plays a role in binding excess copper that would otherwise be toxic. Blood clotting factor V. It contains 1 atom of copper per molecule. This indicates that copper may be required for blood clotting. Copper has 2 oxidation states, Cu+ and Cu2+. Hence, copper is a cofactor for certain enzymes. Copper accepts and donates electrons and is involved in reactions involving dismutation, destroying highly toxic form of oxygen called superoxide ion, hydroxylation and oxygenation. Copper is required for formation and maintenance of myelin, a protective layer covering neurons, nerve cells. Other functions. It has been suggested that copper may have a role in thermal regulation, glucose metabolism and immune function. Thus, copper plays an important physiological role in oxidation reduction reactions, connective tissue formation, iron metabolism, central nervous system, melanin, pigment formation and blood clotting. 10.5.2.6 Chromium. All plant and animal tissues contain chromium. Dietary chromium occurs in multiple valence states. Most of the chromium in the food supply is in the trivalent state, Cr3+. Chromium is poorly absorbed in the intestine. Some of the chromium ingested with the food and inhaled as dust finally reaches the tissues. However, the concentrations are extremely low and highly variable. Chromium is a bone-seeking element and its uptake in bone appears to be rapid. Besides bone, chromium accumulates in spleen, liver and kidney. Generally in human adults, most tissues contain 0.02 to 0.05 ppm of chromium on wet basis. Functions of chromium. Chromium has been shown to be particularly effective in serving as a cross-linking agent for collagen, a protein present in connective tissue. Chromium is a component of a low molecular weight protein called chromodulin which potentiates the effects of insulin, possibly by facilitating insulin binding to cell receptor sites. A symptom of chromium deficiency is impaired glucose tolerance, a result of decreased insulin effectiveness. A number of beneficial effects of chromium on lipid profiles have been reported. Total cholesterol, LDL cholesterol and triglyceride levels have decreased, while beneficial HDL cholesterol and apolipoprotein A levels have increased. Intense public interest has emerged in using chromium as an ergogenic, muscle-building aid. Studies have reported an effect of chromium on body composition. Young men undergoing resistance training and taking chromium supplementation increased lean body mass and decreased fat mass. 10.5.2.7 Cobalt. It is also considered as an ultra-trace element. Various studies indicate that the retained cobalt is taken up by all tissues, the highest concentrations occurring in the spleen and pancreas. The green leafy vegetables, especially spinach, are the richest source of this element and dairy products and cereals are the poorest. Functions of cobalt. Cobalt is a constituent of vitamin B12. You have already studied the functions of this vitamin. It functions as a coenzyme. The B12 coenzymes are called cobamides because of the presence of cobalt. You should revise the functions of vitamin B12 done in the earlier section under metabolic pathways. 10.5.2.8 Manganese. It is classified as an ultra-trace element. Ultra-trace elements are those elements with estimated dietary requirement of usually less than 1 mg per day. The body of a normal 70 kg man is calculated to contain a total of 12 to 20 mg manganese. It is distributed throughout the body tissues and fluids and is not specifically concentrated in any organ or tissue. However, manganese tends to be higher in tissues rich in mitochondria and is more concentrated within the mitochondria than in the cytoplasm or the other organelles of the cell. Accordingly, organs rich in mitochondria such as liver, kidney and pancreas have a relatively high manganese concentration. In contrast, plasma manganese in humans is extremely low. Functions of manganese. Manganese as Mn2+ activates a number of plant and animal enzymes including oxidoreductases, lyases, ligases, hydrolases, kinases, transferase and various decarboxylases. While Mn2+ is specific for glucosyltransferase and xylosyltransferase, other divalent ions may replace Mn2+ as a cofactor in the case of other enzymes. Mitochondrial superoxide dismutase contains Mn2+. Glucosyltransferase links carbohydrate to protein during the synthesis of glycoproteins. There are only a few manganese metalloenzymes, i.e. containing manganese in the structure. These include arginase in urea cycle, pyruvate carboxylase in gluconeogenesis, glutamine synthetase in glutamine synthesis, and superoxide dismutase, scavenger of peroxide radicals. 10.6 Summary. In this unit, we learned that about the different classes of vitamins, fat-soluble and water-soluble. We had a look at their structure, different forms and various biochemical functions. Then we moved on to the study of minerals, macrominerals, required in much larger quantities than the other class called as micro-minerals. Here we dealt with an exhaustive list of these minerals and got to know about their structure and functions. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.